Hello everyone, welcome back to the cell signaling course, and continuing from where we left in the last class, where we were discussing a unique type of receptor tyrosine kinase signaling which involved receptor which is bound to the membrane as well as the ligand which was bound to the membrane. Receptors were called EPH and ligands were called Eph-Ephrin. We saw how it induced a repulsive signaling, it induced a bidirectional signaling, and it induced a phenomena where we had seen the transcytosis happening to remove productive complex of EPH and Eph-Ephrin from the cell surface. So in brief, we saw some very unique things with this particular signaling, where one of the event was ligand and EPH receptor. There is something called cis inhibition, where the receptor bearing cell also has the ligand. So what you notice here is that this receptor is engaged in productive complex with the other cell, but presence of this ligand on the cis surface basically does not activate the receptor bearing cell. So basically in this scenario what happens, the signaling happens in the reverse direction, but signaling does not happen in the forward direction, which is your cis inhibition event. Also we saw how oligomer formation was going on and how the cluster was internalized through transcytosis. We also noticed that the C-terminal tail could be phosphorylated at tyrosine residue and serine residues. So sometimes independent signaling from some other route activates a kinase which is serine threonine kinase, and that ends up phosphorylating these serine residue, which kind of initiates a signaling in the ligand bearing cell. Now if we look at EPH-Eph-Ephrin signaling and think about what all goes on over there, so simply what it can engage in, let's take three scenario. We have been seeing that how the receptor tyrosine kinase signaling is important for survival, growth, proliferation, and migration and so on. So let's take example of ligand, receptor, and you can inverse the scenario here where receptor is like this and ligand is. Generally you prefer EPH-A interacting with Eph-Ephrin A, and similarly EPH-B with Eph-Ephrin B. However, in last class we saw how crosstalk is feasible. So EPH of A type is interacting with Eph-Ephrin of B type and so on. So here this kind of signaling helps in the processes. If I note down, so it inhibits the migration in the cells sometimes which are in the vicinity for some extracellular cues. If they need to migrate, they will be inhibited for the migration. Then sometimes it does the opposite, promotes cell migration, like in case of wound healing. you need cells to move towards the wound and heal the wound. So Eph-Ephrin signaling is involved in that as well. Also, the process can happen in both cell type, the cells which are doing the reverse signaling or the ones which are doing the forward signaling. And by now you would have appreciated that how by different means like cis inhibition or oligomerization, the effect can be magnified or it remains normal. So let's take some example how Eph-Ephrin signaling is shaping the tissue organization or patterning and so on, right? And what are the functional outcome of this Eph-Ephrin signaling? So we'll take example of tissue boundary formation. Now think about a scenario where there are two set of cells which are in close proximity, right? So if I say one set of cell, I'm just drawing as a sheet. The edge which is facing the other type of cells the edge will be expressing, or the cells at the edge will be expressing and engaging in interaction. So if suppose this is the cell which is expressing EPH and this is the cell which is expressing the Ephrin, they will get into the productive engagement and then they will repel. So there will be productive engagement, signaling and repulsion. So basically what will it suggest that this is the boundary region. The cell will not be able to move in this direction and these cells will not be able to move in the following direction. So this kind of sets a boundary between the tissue and they remain in that boundary. That is how the entire tissue boundary formation will come into effect. So you can imagine that this process will be happening during early developmental events. When tissues are being formed, organs are being formed, their organization of tissues is going to be very important, how far they can grow, how small they remain and so on. This is very well designed that what should be the size of a given tissue in developing organism so that it acquires the final size and shape. So that has to be regulated early in the development. So we'll see many such examples where Eph-Ephrin signaling is playing very important role. And here we need not mention which type of EPH, which type of Ephrin, because from tissue to tissue type it will vary. But remember that productive engagement of EPH and Ephrin signaling is contributing to the tissue boundary formation. Let's take another example of wound healing which I just told you. So what happens normally, you have cells, consider this is a set of cells which are present as sheet. These are sheet of cells. And we know that each cell is connected to another cell through some sort of junctions. Cell to cell junction is there. So there will be a cell to cell junction between these cells, right? And these junctions will hold the set of cells as an epithelium together. So in wound healing, we are talking mostly about the surface wound healing, not deep wound healing here, because a lot of cell migration will be happening in both cases. However, it will be easier to understand with the epidermal wound healing, where surface cut has happened. And what will basically do, it will activate. Suppose we say a damage or wound is generated. So the cells which are in between, they now activate. So here is activation of Eph-Ephrin signaling. Eph-Ephrin signaling is activated. And because of this, what will happen? The junctions are internally inside the cell. If this is the junction and this is the cell interior of the two neighboring cells. So internally they are connected to cytoskeleton. So as long as they are connected to cytoskeleton, the movement of cells will not be possible. So Eph-Ephrin activation, what it does, it induces the removal of these cytoskeletal element from these junctional complexes. Thus, eventually what will happen? Now you will have cells, but junctions are not functional now. So you will have the cells which are separated. And these separated cells will now be able to sense the presence of molecule in the environment and then do some sort of chemo attraction and move towards the wound to fill the gap which is created over there. So this is an example of Eph-Ephrin signaling coming to wound healing. You realize that this is becoming more prominent, especially in developed tissues where the damage has happened between the cells which are in the proximity. In the previous case, when we saw tissue patterning or tissue boundary formation, that would have happened in very early developmental processes, but here we are talking about something which may happen very regularly to grown-up individuals. Then there is something called distribution of cells, and here we take particularly the example of intestine. There are different regions in intestine. Within the intestine, there are cells. So if I say this is the intestine region, there are cells which are secretory in nature. And there are cells which are non-secretory in nature. And particularly if we take example of the villi which is present in the intestine. If we take further closer look and we talk about so this is the villi-like arrangement. Now this projected area is basically what we will consider as villi, and here you will have the gland. So the distribution of cells is very clearly understood here, that you will have cells in the boundary region which are actually kind of support cells. Then you can have another type of boundary which could be secretory cell, and then in between you might have a unique cell which is kind of stem cell. So this arrangement is very well defined, that where should be support cell, where should be secretory cell, where is differentiated cell which is no more dividing, where is stem cell which when required can divide and become a support cell or become a secretory cell. This arrangement is very well defined. What defines that? Expression of, suppose we are talking about this boundary. So expression of Eph and Ephrin is something which is defining that where is the boundary for one type of cell which may be involved in the tissue formation, other type of cell which may be involved in tissue function like secretion, or the cells which will be required to replenish if any one of these two cells are not available or die or undergo damage. So this is very important, this is very important process which is regulated by the selective expression of Eph and Ephrin in the cells which are at the junction. So you can have, for example, there are Eph B and Ephrin B, which are known to be expressed in different cell types. The cells which are in the bottom area, where more of expression of or stem cells type cells are present, they express the EPH, and the cells which are here, they are expressing the Eph-Ephrin. So a crosstalk between them. So this is not only affecting, so this would have helped in the boundary formation, as well as it is arranging the cells within the tissue, defining the boundary within the tissue. Previous case which we saw was that defining the boundary between the tissue. Here this is defining the boundary within the tissue, ensuring that a set of cells are clustered together to perform a very specific function. The other process where it plays very important role is in the axonal guidance. So we all understand that when a brain development is happening, or during embryonic development, when neuronal projections are going to different parts of the body, they terminate at a particular place. They are within the brain. If you talk about within the cerebral cortex region, if you talk about the axonal ends, they terminate at very particular position or in a set of tissue, and this is where again EPH-Ephrin plays very important role. So if you consider a set of cell which are already present, and think about them as a cell type A, which are expressing a particular type of EPH or Eph-Ephrin. If suppose a neuronal axonal end is coming here, neuronal cell with dendrites, so its axons are coming to this tissue. They will come here and get into, so if the cell type is expressing EPH, your axonal end is expressing the Eph-Ephrin and vice versa. So when they have similar type of EPH and Eph-Ephrin combination, which can go into the productive engagement, what will happen? The growth cone normally will try to establish contact here. As soon as they see the productive engagement, it will be retracted, which is a kind of signal that this is not the end where you should terminate. So growth cone grows further into the tissue, gets into the productive engagement through EPH-Ephrin signaling, and the repulsive signaling goes on and the retraction happens. You remember that how disengagement was happening, transcytosis, or by cutting the extracellular region of the ligand. So then this growth cone which actually entered the tissue will come back. If it tries 100 of times, it will not succeed because it is getting into the productive engagement and the signaling. Now what will happen? Suppose after seeing multiple attempts, this axon moves in a other direction, and this is another tissue region. What is happening here? You do not have right type of Eph-Ephrin present over here. So this tissue moves into this, does not get into the productive signaling, and repulsion, does what it does, that the axon keeps moving and finally terminates where it is destined to. So axonal guidance is also tightly regulated by EPH and Eph-Ephrin expression. If it is right type of combination of EPH-Eph-Ephrin, there will be repulsion, and eventually it has to go into the other direction where the right type of EPH-Eph-Ephrin combination is not there. Thus there is no repulsion and the axon growth cone keeps moving forward. Then there are tumorogenic outcomes of the anomaly which can happen in the EPH-Eph-Ephrin signaling. You remember we discussed about the intestinal villi region and the cells of the wall. Their clear boundary, suppose one boundary is made here, then in between you have a unique cell which needs to respect its boundary. But for any reason there is a problem in the EPH expression. So consider that this domain is where EPH is expressing. So if EPH is expressing here, it is defining the boundary. If there is a problem which goes on with the EPH because of mutation, some mutation, and or expression of other type of EPH, what will happen now? It will not respect the boundary and growth will happen. Unregulated growth happens and the tissue basically grows. So this region has actually seen overgrowth. And you got a new set of cells at a location where they are not supposed to, because of the mutation which happened in the receptor. This actually can cause cancer, and the situation of APC, adenomatous polypus coli, is something which is reported for these EPH receptor mutant, and you will have a formation of adenoma. Basically, adenoma will be a cancer of cells which have secretory potential within this intestinal lining. So again, respecting the boundary within the tissue is something which was destined by EPH-Ephrin interaction. However, a mutation can actually overrule that boundary and will have a cancerous growth like seen here. Now, if what has been seen that EPH-Ephrin signaling is also playing very important role in something called artery vein plexus formation. If you think about a tissue, if you consider that this is the arterial end and this is the venule end, and in here you have something called plexus. Basically, your capillaries lie here. So this demarcation of in this arterial end and venule end plexus is maintained by the EPH-Ephrin. So what is known is that if you think about a random distribution which is there in the beginning, one of the molecule which has been studied here is Ephrin B2, which is expressing in this region, and EPH B4 is expressing in this region. So when this plexus is developing at early stages, you have Ephrin and EPH distribution like this. Towards the end, when the maturation happens in the plexus, you will see very nice plexus arrangement, and the domains of EPH and Ephrin will be very well restricted, so that you do not have any complication in the mature plexus. So EPH-Ephrin signaling is ensuring that mature blood vessel plexus is forming in the tissues, defining the arterial end and the venule end. So this is where we see that how signaling is playing, EPH-Ephrin signaling is playing very important role in defining that this is your Ephrin containing arterial end and EPH containing venule end, right? So we saw how in many different ways EPH-Ephrin signaling is playing critical role in ensuring tissue patterning, tissue maturation, differentiation and organization and boundary formation. One more example comes from the towards the aorta and vein formation. If you think about early developmental event where you have common precursor vessel, This is defined, if you think about this as a vessel like this, there is, for physiological point of view, there is a dorsal surface and there is a ventral surface. So in this common precursor vessel, there is expression of the Eph-Ephrin and here you have expression of EPH. As this will mature, cell segregation will happen, boundary formation will happen, and basically you will constrict it in between to give rise to two vessels. If you think about the constriction happening along this boundary, so you will have one set of vessels which carry only the Eph-Ephrin and the other set of vessel carries only the EPH. So you will get resolution from the common precursor vessel and you make the aorta region here and the vein region here. If you recollect in the previous slide also, we saw Eph-Ephrin containing part of the plexus became the arterial end and the EPH containing part of the plexus was the venule end. Same thing is happening here. From the common precursor vessel, the dorsal surface which had more Eph-Ephrin was becoming the aorta and the ventral surface which had more EPH became the cardinal vein. So you realize that how EPH-Eph-Ephrin signaling is playing critical role in shaping the tissue, shaping the organism, shaping the organs which are put into the proximity and communicating, making sure that boundaries are formed and to large extent respected. Now if you think about the signaling which happens at the synapse, because I told you an example that how growth cone was moving forward and when it gets into the productive engagement it retracts and moves into the other direction where it does not get into the productive engagement. So basically you will have multiple different ways by which synapses are affected by the EPH-Eph-Ephrin signaling. So one thing is very simple, you can have cell-cell repulsion, different cell types which are present in central nervous system, they will be restricted into different domains because of cell-cell repulsion. We have already seen how growth cone, axons, they are trying to find right zone where they can terminate. What is also known is that when we talk about a glial cell, remember your central nervous system, we always think about neurons, but there are 3-4 times, at least 3-4 times more glial cells are present and they also make strong contact. If you think about the glial cell which makes the blood-brain barrier or the glial cell which is ensuring that neuronal axons are protected. So these kind of glial cells, when they are making contact with axon, EPH-Eph-Ephrin signaling plays very important role in ensuring that whether they will remain in contact or they will come out of the contact. Final thing which is over there is that when we talk about the axons making synapse and its maturation, because synapses are, neurons are plastic, synapses are also very plastic. If there is not enough input, the synapse may just collapse and the synapse may not exist. So for the maturation of the synapse, you will need EPH-Eph-Ephrin signaling. So what you are realizing that there are multiple aspects where Eph-Ephrin signaling is playing a very important role, whether it was intestine, tissue, cells organization in intestinal crypts, whether it was aorta versus vein formation, whether it was plexus formation, whether tissue boundary formation, axon growth cone, and so on, its progression and interaction. So this becomes very important molecule, and what we know is that this receptor, so here CNS, as I have been mentioning, that CNS maturation is very important, and one of the very important aspect of the dendrites making contact with the axon terminal or axons terminating in tissues or axons, the different types of synapses which are forming. All different aspects of axon formation, growth cone formation, its maturation, contacts formation, synapse formation is regulated by Eph-Ephrin signaling. And we are talking about the events which are happening during early development or pre-birth time, as well as when they will be when post-birth an organism is developing more synapses and making more contacts, you will have Eph-Ephrin signaling working over there. And that is why it is not surprising that EPH receptors, they have been studied extensively in several cancers. And different aspects, whether it is the extracellular domain which is allowing it to bind to the ligand, or the domain which helps it to fibronectin type 3 domain which is helping it to tether to the extracellular matrix, or a small region where protease could cut, internally where kinase domain is, or other interactions which are supported, as well as these regions which are proximal to the membrane, the phosphorylation which happens over there. Several mutations have been mapped to EPH receptors, and many have been studied in the lung cancer, and many more have been reported from many other cancers. So it is not surprising that such a molecule which is important for tissue formation, boundary formation, axon growth cone formation, as well as ensuring that how blood vessels are generating towards the end, is subjected to lot of insult through mutations and contributing to the uncontrolled growth and cancer kind of phenotype. We saw how in case of intestinal region, adenoma formation can be affected by virtue of these small mutations in the EPH receptor. So in this class, what we understood is that Eph-Ephrin signaling is involved in shaping tissues and organs, epithelial cell migration towards the wound so that the wound can heal, or intestinal epithelium boundary maintenance. It is because of the Eph-Ephrin signaling. Eph-Ephrin signaling is bidirectional, and it is critical for axonal growth and maturation. The several mutations which have been mapped to the EPH receptor, they come from very different types of cancers. So this Eph-Ephrin receptor presented a very unique type of receptor tyrosine kinase signaling, and it is type of contact dependent signaling, repulsive signaling, bidirectional signaling, showing properties of cis inhibition and many more things. Thank you.